Hello, anatomy. Welcome to Module 16, where we're talking about the senses. Okay, so the senses, so we learned all about the senses when we were in grade school. We learned about the five senses. We taste, we see, we smell, we hear, we touch. Well, it turns out now that we know anatomy a little better, what's really happening is all of these, everything we take in around us is coming in through a sensor of some sort of receptor, and that receptor then sends information to sensory neurons, which then go to the brain or the spine, and then report on what they see or what they take in. So when you're talking about touch, you're talking about a mechanoreceptor. If you're talking about smell or taste, that's a chemoreceptor. That's a receptor that picks up chemicals. There's more going on with touch. Touch is kind of complicated. We also have thermoreceptors, which tell us if something is hot or cold or some variation. We have baroreceptors, which tell us pressure. So whereas a mechanoreceptor just tells us if something touches us, a baroreceptor tells us with how much pressure, because there's a certain amount of pressure that becomes uncomfortable because it can be dangerous. Speaking of danger, we have pain receptors, which tell us if, so these are called the nociceptors, and they are important because what pain is telling you if something could be dangerous or is borderline going to hurt you in a more deeper way. Someone asked me once, what if you're born with no pain receptors? Well, that's actually terrifying. And there's actually a condition where you don't have pain receptors. And parents of kids who don't have pain receptors report that it's a horrifying experience because your kid, one of the things pain does for you is tells you, hey, don't touch that hot thing, you could burn your skin, or don't do whatever it was you just did that made you fall down and hurt yourself, you could break a bone. But someone who doesn't have any pain receptors doesn't get that signal that they are causing themselves damage, so they don't change their behavior. So it's actually very terrifying to not have these pain receptors. They're really important. It may seem like a pain. You can adjust the sensitivity of pain receptors with experience, but that's about it. So electromagnetic receptors take in electromagnetic waves, which is the official way light is described. And they're also called photoreceptors because they are taking in light, photons. In all of these cases, all of these senses are taking an external stimulus or external signals and then transducing it into that action potential that we talked about with the neurons. So they're taking in a signal, they are then converting it to the electrical signal we see in an action potential, and that's going to release a neurotransmitter. Okay, so you see something, that light hits your eye, it hits the receptors in your eye, which then send a signal to the part of your brain that's taking in light, in the whatever part of the brain it is that's relevant, and then that releases a neurochemical that says, oh, that's a kitty, or whatever it is that you're looking at. In all cases, this ion permeability thing has to do with, that's just another way to talk about an action potential. And a neurotransmitter release. So NT means neurotransmitter. It's literally exactly what this is just saying. Adaptation occurs when receptors become less sensitive. So this is where if you're wearing clothes after you put them on the first time and you stop paying attention, you stop feeling them. You don't notice, am I wearing underwear today? Well, if you start to think about it, you might start to notice it. But if you're not paying attention, your body's like, well, I already know I did this. This signal has already been received. I don't have to worry about it anymore. You see this in other senses. We're going to talk about like with smell, with taste, etc. We're going to come back to it. So adaptation is when a receptor becomes less sensitive. So what is happening with touch? So touch happens through these guys, these very specific kind of mechanoreceptors. And I'm not so concerned with you knowing what a Meissner corpuscle or a Merkel's disc is, or a Peckinpah corpuscle, etc. What I do want you to know is that there are mechanoreceptors that tell us if touch is happening, and then there are mechanical receptors, which are also called, these are specifically baroreceptors. Let's put that in there. Receptors, which tell us pressure, and they're two different kinds of receptors. Okay, so touch is complicated. It involves, is touch happening at all, which is a mechanoreceptor, how much pressure is being applied, which happens through a baroreceptor, and is it something painful? So this is happening through a nociceptor. Okay, so these are just nerve endings that don't really connect to anything, and they become sensitized. Okay, so you stimulate them, and then they just send a signal because something's happening. So when you get a paper cut, the reason why they hurt so badly is because you've cut through those nerve endings, and now they're totally free, and then they kind of the flap of skin that you just cut through in a paper cut closes really quickly, and then those nerves just like start touching each other a bunch, and they can't stop touching, so it hurts really badly. So a somatic versus a visceral pain, remember soma means body. So a visceral is an organ pain. So this is something happening basically anytime you're aware of some part of your body, just out of nowhere, something bad is happening. So if you have a somatic pain, then that means you have some kind of external signal telling you, hey, you got a cut, or hey, you have a bruise, or whatever. But visceral pain is going to be coming from nociceptors inside your organs that say, hey, I have a stomach ache. You shouldn't have ate that gross old sandwich, or whatever, that tells you whatever just happened, never do it again. In cases like let's say if you have appendicitis, you have pain in that part of your body, that's telling you something is happening, and your body, in the case of appendicitis, doesn't know what to do about it, and that's why we have to have surgery. But in other cases, if you have pain, it's basically your body saying, something is happening, there's an inflammation, there's something happening here that's not normal, you need to be aware of it and either take it easy or fix it. There is another kind of nerve ending in the skin, which is the ones that tell us where our body is. So this is proprioception. We talk about this a little bit in the lab. So this is where I know exactly right now where my toes are, even though my laptop is on my lap and I can't see my toes. I know where they are. I know when I'm walking, I know how to walk, I know where to put my foot, I know if I close my eyes, I can stumble around. I'm not worried about where my hands are. I'm just worried about what they'll run into, right? So this is actually a really big deal. And we know through disease states where the part of the brain that contains this kind of receptors, or sorry, not the receptors, but the neurons, if you lose that part of your brain, you can become paralyzed without even having a reason other than just your body doesn't know where something is. And so there's a case that Oliver Sacks talks about where he had a patient come in who slowly became more and more paralyzed until she realized, like, someone sat her up in bed once and she looked at her feet and suddenly she could move her feet again. And so she realized as long as she was looking at a part of her body, she wasn't paralyzed. And so she lost her proprioception. But as soon as she could see something, visual sense is so important to our brains that suddenly she got that sense back. And so it's a very strange but really important sense. And we don't really talk about it when we talk about the five senses, but we actually have way more than five senses. The sense of pain, the sense of knowing where your body is, a sense of balance, all of those things go beyond the simple five senses. That's about all we'll talk about with this. That's complicated enough. I think that's enough things to memorize. Remember, you don't need to know these fancy words, but you do need to know mechanoreceptor, baroreceptor, nociceptor, and it's good to know proprioception. Let's talk about the eye. So the eye is all about vision. We have pretty decently sized eyes. The parts of our brain that deal with vision are very big because we're such visual organisms. So there are 6 muscles that control the eye. We've got some, and if you think about it, we want the eye to be able to move in 3 dimensions. We also want it to be able to squish a little bit because it's important. We'll talk about with nearsightedness and farsightedness. It has to do with how we focus on something close and far. The eye, let's go. So looking just at the external anatomy, the lacrimal gland is very important. It produces tears. This is not so you can cry about how much you have to learn from anatomy. This is so you can wash out your eye and also provide nutrients to the cornea. So the cornea is the outer part of the eye here. It connects with the sclera. It's continuous with the sclera. And what it does is it protects, it's transparent, it doesn't have any blood vessels because it needs to be as transparent as possible, and it connects the back of your eye to the front of your eye, sort of. I knew somebody once who went running when it was negative 20 degrees outside. She was really careful to cover her entire body, except for her eyes. And what happened when she was running, her eyes were cold enough and being hit with wind that was so much colder that she actually froze her corneas off of her eye, and they popped right off. And she had to wait either for a transplant or for her corneas to grow back. So they can grow back depending on how you damage them. And you can also scratch your cornea if you've ever put in contacts badly and ended up with a scratch. They do grow back, but it's definitely not pleasant to have to grow back your corneas. If you're an organ donor, a cornea transplant does not require matching. It doesn't require you to match blood types when you do an organ transplant, like you do with other organ transplants, because there's no blood vessel, and it's all about the blood vessels when you're talking about organ transplants, which I'll talk about later. So the lacrimal gland here is releases through the lacrimal duct. That's what's going on here. When you if you're, let's say you're crying, if crying happens, crying is a completely natural human response. Don't, people do it to different amounts. It's nothing to be ashamed of ever. Um, if you've ever noticed your nose gets really runny, it's because your sinuses fill up to help push more water through the lacrimal glands. Well, then you've just got a whole bunch of fluid up in your sinuses also, and they start flowing through your nose as well. Also, when you are cycling the water that's going through your eye just on the externally, it goes back through the lacrimal gland. It can go through the lacrimal duct, and then it empties into your sinuses and leaves through your nose. So that's delicious. Let's take a look at the eye if we cut through it this way. So if you were to look, so here's that cornea that we're talking about, okay? So the part of your eye that's colorful is the iris. Your pupil actually doesn't exist. It's a hole, okay? Your iris constricting and dilating is what causes your pupil to get bigger or smaller. And what it's doing is it's allowing more light to hit the lens. And then as light hits the lens, it gets bent and hopefully hits the retina. So the retina is the part of the eye that actually has the photoreceptors. So that's the first thing that light hits as it goes through the pupil. If it hits any other part, if it hits the iris, it bounces back. If it hits the cornea, it can go through because the cornea is completely transparent. The cornea is also continuous with the sclera, which continues this way. You'll notice there's no blood vessels in this entire part, but the only part that you actually see is the cornea when you're looking at somebody's eye. The whites of someone's eye, I guess you're just looking at the cornea. And the ciliary body, that is what helps the lens focus. And so what's happening with this is the shape of the lens determines where the light that's coming in through the lens is going to cross and then create an image. And that has to then, and we'll actually get to that in a second if we go here. Okay, so if your lens is here, so light's coming in this way, the shape of the lens, the image is going to happen at the focal point, okay? So, well, I guess that's not true. An image, that's where they cross. And so if you were to follow a single line, boop, that's where an image is going to occur. So you're going to follow that line until it hits, okay? So if you're looking at this person, you're going to have light come in. Oh, the lens bends the light, and it's going to go this way. And you'll notice a lens always inverts an image. So when we actually see something, we see it upside down. But our brain says, oh, I can fix it, and it flips it over. So we never actually interpret things upside down. Our brain flips it for us so that we don't have to. It gets weird. So about the ciliary bodies, that's this muscle and this muscle. So if you were to change the curvature of this lens, you change where this flip happens and where the focal point happens, okay? So if you were to pull on these, if you contract these ciliary bodies, the lens gets less curvy, it straightens out a little bit, and that causes you to be able to see really far away, or well, it helps you see far away. So if you think about, if you look at your hand very, very close to your face and you focus on it, you kind of feel something happen in your eye as you focus, whereas if you then look beyond it and focus on something on a wall that's far away, that also, you can feel something different happening in your eye when that happens. And what's happening is those muscles are contracting and relaxing. So that's the ciliary body muscles, and we'll talk a minute about that. We also have, so the other muscles in the eye that we're talking about here just help you look up, down, and left and right. That's all we're going to talk about with those. What else is happening here? The coracoid, or the choroid, sorry, is just the blood, well, it's just basically another protective layer in the eye. I'm not really concerned about that. The optic nerve, however, is very important. That's where all the signals that are coming from those photoreceptors are going to be going and then summing in that nerve and then going to the brain. So any light that hits right here does not make an image because there's no photoreceptors here. It's just an empty space that then is where all the nerves go. So we have a blind spot in our eye, which our brain tries to make up for. We also have this fovea. It's another no, that's actually, I think the fovea is where we have the most photoreceptors because you have a lot of surface area here. We also see really well in our periphery, and like if it's ever really dark and you're looking for something that's light colored, if you look to the side, you'll be able to see it better, and that's because we pick up more light in our periphery than we do, or we pick up the most light and make the most sensitive image from our periphery. And the thought is that evolutionarily, if you're looking forward because you're worried about a predator and one's coming up from the side, well, you need to be signaled to that really quickly, and that could be a reason why peripheral vision needs to be so sensitive, at least to motion. Another thought in that if you've ever gotten up and left a room and forgotten why you went into another room, there's something that's been scientifically proven that, or at least shown, that when you walk through a doorway, you're more likely to forget something. And it could be that you're triggering that peripheral sense, and then suddenly your brain's trying to be more aware, and it forgets what you were originally just kind of idly thinking about. And so that could be part of that as well. Okay, so the choroid coat is what's bringing in the blood vessels that are going to keep the retina healthy and fed, okay, because those are neurons and they take a lot of nutrients. You also have blood vessels coming in along that same optic nerve tract, and what they do is they're providing blood into this region. They also are helping to cycle this fluid that's inside our eye, our aqueous humors, which I'll talk about in just a second. We can actually divide the eye into two parts. We have this back part and this front part, both of which are filled with fluids. It's a different composition. You can get floaters if you ever look at something and you see stuff kind of floating in your eye. If you get floaters here, you can see them. If they're here, though, you don't really see them. And what all floaters are is just parts of like blood vessels that have broken down and not been cycled. We all have them. It's not a weird thing. They're not, bacteria trying to destroy you or anything. They're just chunks of stuff that happen in biology. So it's not a big deal. So you don't see them if they're really close to your lens. As they get closer to the receptors, you start to see shadows. So as you get older, you see these little tiny shadows. If they're in front of the lens, you see through them. And you can see like they're clear things. If there's certain kind of diseases where you just start to get a whole lot of buildup of black material, and that can be problematic. And that's one of the many reasons to go to your eye doctor. So, let's see. I already told you about this. Let's keep going. So, okay, so let's talk a little bit about light. We kind of already talked about that. Let's talk about if the lens is not focusing that point of focus on the right place, you don't get the image you're going to get a blurry image. So anytime so let's say it's a normal eye, you get the bending, it's going to hit whatever that focus point is. You want that focal point to be right on the retina, because that's going to be giving you the light that is going to stimulate the photoreceptors to then tell the brain what you're seeing. So if they're actually hitting and focusing a little bit closer than that, then they're going to spread out again, and then you're going to get a fuzzy image on your retina, and that's nearsightedness. So if they're focusing closer, it's because your lens is curvier, and so there's thought that this might be caused by if you were like me as a kid and you'd stay up way too late reading books or comics when you were supposed to be sleeping, so you'd have to do it in the dark and focus really, really hard, really, really close. The idea is that you were squishing your lens for too long, and then the ciliary body can't unsquish. I don't know if that's actually true. It could be urban legend, but that's the thought there, is now it's way too curvy and squished and you can't undo it. So this is technically it's called myopia, but this is nearsightedness. So you can see close, but not far. Like things get blurry as you look further away. And it can get pretty intense. Like my myopia is such that things start to get blurry about a foot from my face. And then they just never resolve unless I get closer or if I put glasses on. The opposite of this is if the lens is not curvy enough and the point of focus would actually be beyond the retina. And so in that case, things that are farther away look clearer than things that are closer. Okay, so that's the difference here. So hyperopia, or farsightedness, is when you can see things far away but not close. Myopia is when you can see things that are close but not far away. And then just normal vision is when it's, that would be, something that is 20/20, some person who is 20/20, what that means is you can see things at 20 feet that someone with normal vision can also see at 20 feet. If you have, I think it's 20/10 vision. That means what a normal person sees at 20, you can actually see we can only see at 10 feet. So that would be someone who's nearsighted. If you have 10/20, what I think that means, and I could be wrong, is that you can see at 10 feet or what someone sees at 10 feet, you can see at 20 feet. So you actually have better than average vision. And yeah, let's keep going. So if you have astigmatism, that means the cornea is warped. And so it's not just too curvy or not curvy enough. The whole thing is warped, or it could be the lens is warped, or it could be you have some muscle condition that's a problem. And then it gets a little bit more complicated. So for this class, just know that an astigmatism is where you have a warped cornea or lens. And it's beyond this. It gets a little different. Okay. So it's talking about how there's cavities in the eye. These are not eggs. This is actually a cow eye that's been separated. So if you look here, so we can see that, so this is a glass model, and so it's not perfect because this line does not exist in eyes, obviously. But if you look at a cow eye and look at it this way, so this is the iris, this part, the colored part. This would be the pupil that you're looking through. So it doesn't look black because you're looking at whatever the surface is. You're looking through the lens at the surface. This side of the eye, so this side of the eye, is filled with aqueous humor, okay? And this just has a lot of nutrients for the cornea that's being supplied sometimes by the lacrimal glands, sometimes by the choroid vesicles, okay? The vitreous humor is in the back, and it supports the retina. So you have a separation of the cornea and the lens getting nutrients here, and then the retina getting nutrients in the very back. And you'll remember this cavity is so much bigger because you have to allow for that light to focus on the retina back here. So that's important. Okay, so what is happening in pupil dilation and constriction? So we take in the signal that there's a lot of light. Oh my God, there's so much light that I need the pupil to constrict so I don't burn my retina with too much light. And so then you have these circularly arranged smooth muscle fibers in the pupil, and they contract, or sorry, yeah, so those muscles contract, and it's circularly closes the pupil, much like a sphincter does, okay? If, on the other hand, there's not enough light, that sympathetic motor fiber will then say, hey, I can't see anything. So sympathetic means it's part of the system we can't control, so it's what your body does automatically. And so it starts to open up that pupil so you can take in more light. Now, there are other things that also cause this dilation of the pupil, this opening of the pupil. So there are drugs that do it. There's drugs that your eye doctor gives you to make sure that they can see back into your retina to make sure it looks healthy. So that's why you get your eyes dilated at the eye doctor. There are also just there are recreational drugs that will do this too. Also, just naturally, if you are oftentimes when we are scared, or if we are emotionally aroused in other ways, like through either sexually or not sexually, but like just if you're excited about something, your pupils will also dilate. And the idea is you are in an excited state, and so your body's like, we better take in as much light as possible so we can figure out what's happening. And that is another way to get this pupil dilation to happen. Okay, that's all you need to know. So what about, PowerPoint doesn't believe in words, but, whatever. These things, no. Celery, okay. So what about when you're focusing? What's happening when you focus? So we have these ligaments, these suspensory ligaments in the ciliary muscles that, again, like I talked about, so if you contract them, you cause the lens to become less curvy, and so then you can see really far away. If you let them relax a lot, let's see. So if the ciliary muscles are relaxed, but then these suspensory ligaments are contracted, then you can see far away. So the lens gets really thin. And really the thing to remember here is if the lens is thin and not as curvy, you can see far away. That focal point is farther away in the back of your eye. If you make it more, if the ciliary muscles contract and the lens becomes thicker, so it gets squishy, it becomes more curvy, you focus much more closely. Okay, so we can do this a little bit. This helps us see near and far. There are a lot of animals that can actually do this much, much better than we can. If you think about whales and other aquatic animals like turtles or fish, they can see both, some fish can see both above and below water. We can't do that because there's such a change in the way the light is being bent through the water versus air, that in order to do that, you really have to squish out an eye in this way. And so if you see like a dolphin looking above the water, they look like they have kind of bulgy eyeballs. But when they're underneath the water, they have very flat eyeballs. And so the difference is their muscles that are controlling the lens are much more powerful than ours are, so that they can see both above and below the water. Okay, that's what I want you to know about that. Not so much. So know that the ciliary body muscles and the suspensory ligaments are what control the lens curviness, but definitely know that when it's long and thin, you can see far away, and when it's thicker and curvier, you can see really close. You can focus more closely. So specifically, when I talk about the retina, on what neurons are happening in the retina, those photoreceptors, what neurons are in there, we have two kinds. We have rods and we have cones. And they're called rods because they are long and thin. And then we have cones because they look like cones. And so what is happening, and what's interesting is our retina is set up, well, we're going to get to this in a second, but our retina is set up backwards. The part that gets stimulated is here. So this is where the photosensitive chemicals are that say light is happening. So rods tell us just black and white. They're very, very sensitive to the amount of photons present. So these are going to be the ones that are active when you're night adapted. So if you, and I talked about this in lab, but if you cover up one eye and not the other and let it get really, really adjusted, and then suddenly turn off all the lights and then take your hand off of that eye, you'll see a really big difference in how well your eye adjusts to darkness, okay? On the other hand, and it's, by the way, this is thought to be part of why pirates wore eye patches, but also pirates wore so the idea being that they could be on the top of the deck in the sunlight where there's lots and lots of sun, working with an eye patch. Like, let's say they put an eye patch on their left eye, their right eye is open to the sunlight, and then they go below deck where it's really dark, and then they move their eye patch over to the other side, and all of a sudden the left eye that had been covered up by the eye patch suddenly can see really well in the dark because it's dark adjusted. But then the other side, where they were experiencing a lot of light, they don't have to worry about that dark blindness because that eye is now covered. Okay, so what's happening so rods detect black and white, just if light is present or not. Cones, on the other hand, detect color. There's a whole bunch of these in that fovea, that little pit that's very, very sensitive. And we actually only detect red, green, and blue wavelengths of light, but it's the amount of each one of these that gives us all the other colors, okay? So if you have red and blue, according to Art, you should have purple. I think that is what happens in this case. If you have red and green, according to Art, you should have brown, but according to physics and biology, you actually get yellow. And so it's very weird. Don't worry too much about that. But do know, so rods are detecting just the amount of light present. So it tells you in the dark. So basically, rods are really active in the dark. So they're really sensitive to that. When there is light present, so especially when there's a lot of light, you're mostly dealing with your cones, and they're telling you the different colors of image. You can get a better resolution of like very specific things. So animals that have really sensitive vision, like hawks, they have some of the best vision on the planet that we know of, they have basically just cone cells, and so many of them. They like way more than we do. Whereas animals that are only nocturnal and they never experience sunlight, they have all rods instead, and they're just very, very sensitive to the presence of light and darkness, but they don't really care about color because it's nighttime, and they're not really worried about if, something is blue or purple. They're worried about if something is present or not present. Okay. I'm not going to worry about this. This is what specifically, if you're interested in looking at the chemicals happening in the photoreceptor, it's happening through rhodopsin and opsin. I don't care about this. Let's move on. But let's kind of look at this. So the weird thing about the way our retina works is that it's backwards. And it's one of these parts of evolution where, well, it doesn't not work, so it's just going to keep happening. So the retina is composed of those photoreceptors, the rods and cones, okay? So here's a cone, here's some rods. I guess we'll say that. Maybe these are rods and that's a cone. I guess that's what it's supposed to be. These are rods and that's a cone. And the idea is the lens is on this side and the light is coming in this way, but the light then has to go all the way through all these neurons without hitting any and being absorbed, and then stimulate these. So our retina is set up completely backwards, okay? So in the case of the octopus, where the eye evolves completely independently, their retina is set up the other way. So the very first cell that gets hit by the light is the cell that's actually sensitive to light. So our neurons don't pick up light. Our neurons are waiting for a signal from the rods and cones. So if there is a green wavelength of light coming in, it has to go all the way through, dodge all the neurons, and then bounce off the back of the retina and then excite that green cone cell. Ridiculous, but whatever, it doesn't not work. In the case of animals that are nocturnal, and you may have seen this if you have a cat or a dog, they have this shiny coat back here that's called the tapetum lucidum. They have a shiny layer, and that's why their eyes flash like that red or green or yellow or maybe sometimes blue flash that you see in your pet's eyes. Okay, that is the reflection of that crystal layer in the back of the eye. We have it somewhat. You see it a little bit in red eye. Some people say you're actually just seeing your optic nerve, which is not what you're seeing. You're seeing the blood in your eye. You're not seeing that. You're seeing the back of your eyeball, which is mostly a red color with some shiny crystals in it to help bounce those light rays back. But the most intense crystal layers happen in nocturnal animals. Okay, but it's very important to know that the photoreceptors, those rods and cones, are what are transducing or translating that signal from light into the release of a neurotransmitter. And so those neurons are then carrying the signal to the brain through the optic nerve. Okay, so light comes in, it stimulates the rods and cones. The rods and cones are like, hey, something happened. And they tell the neurons, which then all go to the brain to tell you what you saw. Okay. So this is kind of taking that idea. Interestingly, so we are binocular. We have two eyes. There is an animal that is sexnocular. The mantis shrimp has 6 pupils, and so that means it actually has a lot of overlap in how its eyes work. It's very complicated. Scientists are still trying to figure it out. But interestingly, while our brains are very large and very intensely interested in vision and interpreting vision, the mantis shrimp barely has a brain at all. So it's kind of weird to think about, well, why does it have so much interesting eyes, so many interesting eyes, but not much processing power? Whatever. So the interesting thing here to notice is there is an overlap in what this eye can see and what this eye can see. However, there's a blind spot, okay? So we can't see right close to our eye, but that's okay because our nose is there. But then you'll notice there's a point here where you can actually see your nose, but because of adaptation, our brain stops paying attention to it because it's like, yeah, I know my nose is there, so unless you want me to actually look at it and pay attention, I don't care because it's just I don't need to know about it. Let's see. There's also a part where only this eye can see and a part where only this eye can see, but our brain just kind of puts it together into one image, and this is how we can see how depth works. You have to have both of them working to get depth, okay? You might think, well, but I can cover up an eye and still reach for something and know where it is, or and grab it, but that's because you already know how far away it is because you know how big it is, so that's not a good excuse. The other interesting thing to note here is that when all those neurons that are coming from the retina, from those optic cells, those rods and cones, when they go through the optic nerve, they split in what's called the optic chiasm. Important! The optic chiasm is here. And so you'll see half of the neurons from this eyeball go to this side of the brain. Half the neurons from this eyeball go to that side of the brain, and half of them stay on the same side. There's a lot of interesting reasons for that happening. We're not going to go into it in any great detail, but it is an interesting fact that you're not just seeing everything with one side of the brain and not the other. That's all I want you to hear. There is one more eye test that you do when you go to the eye doctor, where what they're doing is they're looking at how your optic chiasm is working. They're basically looking for blind spots, because if you have any blind spots in your optic, like your optic chiasm is not seeing something that's here. So what's actually happening is part of your brain could have a tumor in it that's pushing down on the chiasm, which is then causing you not to be able to process light coming in. So that's another important eye test you do when you go to the eye doctor. Last thing about eyes, we only have a blue, a green, and a red cone cell. So again, if you think about the mantis shrimp, they actually have 12 versions while we only have three, so they can see, oh no, it's the mantis shrimp, it might also be the octopus. They have 12 cone cells, so they can see even more detail when it comes to colors. There are some people who have a fourth cone cell, and they're called, I don't remember, but it's hard for then people to be like, well, how does someone with four cone cells who can see even more detail in colors, how do they see compared to other people? Well, how do you describe red to someone? You can't really describe, well, red looks like this. I mean, it's a very personal experience, right? So if I see this as red, I know what that means to me. I also know that I've been told my whole life that this is red. But what if what I'm seeing as red is what you actually see as purple or something? Like, we have no way of knowing how your brain personally interprets things, which is interesting. But we can say, I know that when you're stimulated by this wavelength of light, this cone gets activated, and your brain will then interpret it however it interprets it. Uh, so when you see all the colors of light, and by the way, there are only six, there are not seven. Newton decided to make one up because he didn't like the number six. So Newton's the one who invented the Roy G. Biv. Well, he's the one who discovered that rainbows always happen in the same colors. He discovered that white light contains all of the colors of the rainbow. And if you think Roy G. Biv, so red, orange, yellow, green, blue, indigo, violet, there's actually no indigo. So he just didn't like the number six. He liked the number seven. So he pretended, and in his paper where he's describing this, he's like, sure, there's a seventh color. It's between blue and purple, and I call it indigo. But it doesn't actually exist. It's a blending of two different colors. We can see all of these colors and all the ones in between based on how much each one of these cones is activated. And we don't just have one of each, we have millions of each kind, just like we have millions of the cone cells. That's enough about vision. Let's talk about hearing. So hearing is a very different process because whereas light is an electromagnetic wave and it exists, it can travel through space, it can travel, it only travels in straight lines, and it can't travel around corners. You have to have a mirror for that. Hearing is a pressure wave, so sound is a pressure wave, and we interpret it in a different way. Well, it gets interpreted differently through our neurons. So when you look at the apparatus for hearing, the ear, we have the outer ear, the middle ear, and the inner ear. So just like any other part of our body, if you have some way to get into the body from the outside, it's going to be coated with mucus and hair. So that's why we have hair inside our ears and we have earwax, because that's to make sure bugs don't fly in there. Very important. Or bacteria or viruses or worms or whatever. You don't want stuff getting in your ear. Because once you get to the middle ear, that's when you hit the eardrum or the tympanic membrane. Very important. The tympany membranes is just the plural version. That's when you're talking about two of them. The tympanic, let's see. So if you want to get very specific, where the inner ear, the outer ear is the pinna, which is this part. This also contains Darwin's tubercle, which is this little. Some people have a very pronounced bump right here, and that is really this part of the ear is the part of our ear that used to be. If you compare it to like a cat's ear, it's the pointy part of the ear that controls directionality of the ear, but since we don't move our ears anymore, that kind of has been lost over time. But you can see some people have a very pronounced bump here, whereas others don't. It's just an interesting evolutionary throwback. We have the concha, which is kind of this little part here, the endpoint, and then the auditory meatus, which is the acoustic meatus, sorry. Okay, in our lab, we call it the external acoustic meatus. This calls it the auditory meatus, whatever. Either one, that is the tube that leads from the outer ear to the middle ear. So that's the external acoustic meatus or the auditory meatus. Okay, so we have a sound wave, it comes in, it gets funneled by the ear into the meatus, okay? That sound wave comes in, it vibrates the tympanic membrane or the eardrum. That vibration then vibrates these three bones. So these are our smallest bones in our body. You may have heard them as the hammer, the anvil, and the stirrup, but technically it's the malleus, the incus, and the stapes. And so what they do is this vibrates with that sound wave. That then vibrates this bone, which vibrates that bone, which vibrates this bone, the stapes or stirrup. Then we are no longer in the inner ear, so you have all of this vibrating at whatever sound wave frequency is coming in. That then vibrates the inner ear, and that is, we have this cochlea, okay, this whole thing. This tube, by the way, is not involved with hearing. This is a eustachian tube. This is involved with equalizing internal pressure. So when you have to clear your ears, if you're going up and down in an airplane or when you're driving over hills, when you move your jaw, you're adjusting the pressure that's then being pressed against the tympanic membrane and letting that kind of adjust itself. I'm thinking about yawning, and now I really want to yawn. If you're yawning too, it means you're not a sociopath for one reason, because they tend not to be able to pick up people's yawns, or psychopaths. Anyway, one of those two. But it's a totally social thing to catch someone's yawn. But one of the reasons why we yawn is to equalize this pressure. There's other reasons for yawning that we're not going to get into. Okay, we're going to talk more about the cochlea in a second, but so our sound wave has come in, it's vibrated the tympanic membrane, all three bones, and now it's vibrating what this bone is sitting on, which is the window. There's an oval window here, or no, the round window here, one of the windows, we'll get to it in a second, which then vibrates all the fluid inside of the cochlea, which is the snail-looking guy. Okay, so let's look at what's happening. Okay, I kind of already said all of these things. So the auricle or the pinna is this part. We have a sound wave that comes in. It gets funneled in by the concha. The external auditory canal or meatus is just the tube. And then we get to the tympanic membrane or the eardrum that also is going to be vibrating as that sound hits it. In the middle ear. Okay, so we've now flipped this over so the ear is on this side. Here's the eardrum. It's vibrating because sound hit it. That causes the part of the malleus that is touching it to vibrate. That vibration then causes the incus to vibrate, which causes the stapes to vibrate. Okay, those are just the bones that connect from the eardrum to the cochlea. Cochlea. You might think, why is it 3 bones instead of 1 bone? Well, this is much more sensitive. So apparently it just works this way. Like we're talking about the equalizes. How about equalizes? The eustachian tube just helps you equalize your internal and external pressure, and it drains into the back of the nose, so that's fantastic. There are muscles attached to the ossicles, these guys, and basically it's controlling how sensitive you are to hearing. So if you're kind of tuning out background noise, your muscles are relaxing and not letting these vibrate as much. But if you're really trying to pay attention to a sound, these muscles are going to be really they're going to be amped up, they're going to be contracted so that you can have as much vibration here as possible, so you can get even better sound. One of the ways we lose our hearing as we get older is these muscles lose tonnage. Okay, so this is what I just said. So we have that tympanic membrane vibrates this bone, vibrates that bone, vibrates this bone. And there's those muscles that either get really taut, they get really contracted to be very sensitive, or they relax a lot because you're not really paying attention. All right, so in the cochlea, so here is the stapes, which is connected to those other bones. So the stapes is vibrating with whatever sound wave is going on, okay? And that's vibrating part of this, it is the oval window, the part of the cochlea. Well, it's basically pushing back and forth and creating a pressure wave, a fluid pressure wave within the cochlea. Okay, so if we were to unwind this and look and see what's happening, depending on how far you get in the cochlea, depending on how much vibration there is, that's going to be an increase in the frequency of the sound you're listening to. So when that happens, so if you look at this way, so the further along here, it's going to be the deeper it is. So the higher the frequency, or the higher the sound, the pitch of what you're listening to. So the higher note that you're listening to is going to be sensitive. It's going to be heard first. The lower pitches are going to be able to travel further, and they're going to, like, our very lowest pitch is going to be able to hit here. And so basically the longer the frequency of the sound wave coming in, the further it goes through the cochlea, okay? And so what's it doing when that sound wave is coming in and it's vibrating the fluid inside the cochlea? Well, how does that vibration then go to our neurons? I'm glad you asked. So within that, if we then cut a cross-section of this going this way and we look inside, you have this vestibular canal and the tympanic canal. That's the two sides of this. So here's the tympanic canal, here's the vestibular canal. I guess it's on the other side. So this is tympanic, this is vestibular. So when you look inside there, you'll notice there are all these weird little cells going on in the organ of Corti. All I really want you to know is inside these canals, we have these cells called hair cells. Okay, so within that organ. And what those hair cells do is as they're vibrating with that sound wave that's coming in, so that tympanic or the tectoral membrane that's being vibrated by the fluid inside the cochlea, they're being vibrated. It's like kind of like an electric toothbrush vibrating its bristles on your teeth, so you can hear through your teeth, basically. Depending on what part of the hair cell is being vibrated, that sends a signal to the brain saying, I heard this sound and I heard it this many times, and then that is how you translate sound to, that's how a sound wave gets transduced to what your brain is interpreting as sound. So let's see. The movement of the perilymph, which is the fluid inside of the cochlear canals, moves the membrane, that basal membrane, which is inside. So the basal membrane is going to be the bottom membrane of each one of these, okay? So the membrane inside of the canal is going to be vibrating, and depending on where it is within the whole cochlea, you're going to vibrate at certain frequencies. That then pushes those hair cells. And when you're pushing those hair cells, you're transducing or bending those hairs, and you're depolarizing that neuron, which causes a signal to be sent. Okay, so again, you have the sound wave comes in, it vibrates the fluid inside of your cochlea, inside your inner ear. That fluid vibration is going to vibrate this membrane, which then shakes those hair cells, which then send the signal to the auditory nerve, which is what, so like the optic nerve takes light signals, the transduction of the light signal. The auditory nerve takes the transduction of your, the sound wave that you're listening to. Okay, what did I just say? That's a bunch of crazy things. So here's the order of what happens. Hint, maybe know this for the test. So you have some sort of pressure wave, a sound wave, that hits your ear. Okay, it goes in through the external auditory meatus, or the external acoustic meatus, or your external canal. It vibrates that tympanic membrane. Okay, so it goes into the ear, it vibrates the eardrum, the tympanic membrane. That then is going to cause the vibration or mechanical motion of your ossicles, or your ear bones. Okay, so blah blah blah, these are vibrating with the same frequency that sound wave was vibrating at, that then vibrated the tympanic membrane. Vibrate. That then vibrates the oval window. The oval window vibrating creates a fluid pressure wave. So you get this pressure wave inside of this fluid, the fluid inside of here. That fluid pressure wave vibrates that membrane that's sitting on the bottom of each one. And then that bends your hair cells, releasing neurotransmitters, which then sends a signal to the brain. Okay, very important. Study this slide. If you're listening to this, you know this will be on the test. All right. So there's actually something else that happens in the cochlea. If you go up and you look, so the part that processes sound is the snail part, but then this crazy looking thing, these are called the semicircular canals, and what they do is they deal with balance and equilibrium, because another sense we have is where we are in space. So am I standing upright? Am I facing downwards? Am I moving forwards or backwards? Where am I and what am I doing? So there are these guys. So the inner ear is the cochlea, the vestibular system. The vestibular system is the balance section here, okay? And that contains a utricle, the saccule, and the semicircular canal. So let's investigate those. So when your head is a certain way, we have inside the utricle and saccule, you have a whole bunch of more of those hair cells, okay? Yay, that's great. Hair cells tell us sound. Well, they do when they're inside the cochlea on the other side. When they're in the utricle and saccule, what they do is instead of being moved by a membrane, you have all these otoliths, or little rocks that sit on top of a gel. Okay, and when you move your head forward, all those rocks kind of go tumbling, and they hit the cells on the other side. Oh, well then that stimulates those hair cells, and it says, hey, you've moved your head forward. That's interesting. Okay, so if you move your head if you jump up and down, the hair cells kind of take a second, or it's not the hair cells, the otoliths take a second to go up and then down. So you get a kind of weightlessness feel for a second because those otoliths are not putting pressure on the gel, which is not then putting pressure on the hair cells. Okay? If you are drunk, so if you're drinking to the point that you have less than, or maybe if you have 1% blood alcohol, which is actually a very high illegal state to be in, what that happens then is the fluid inside your ear gets just slightly diluted enough that these ossicles are moving faster than your brain knows how to process. So if you've ever been in that state, you turn your head, it takes a second for the otoliths normally to catch up, but in the case of when you're drunk and this is a much less dense fluid, these move a lot faster than you expect, and your brain doesn't process it right, and then you fall down. So that's part of why you fall down a lot if you're drinking too much. Other drugs also do this. Okay, let's keep moving. The semicircular canals, on the other hand, they have these cupulas that actually sit over the hair cells. So instead of a rock, they have the cupulas. So where the utricle and saccule are telling you how you're moving in space, as far as are you looking up, are you looking down, have you moved very quickly or not, just based on where these rocks are, which is kind of a very simple system. The semicircular canals, you have one in each direction. So you have one in the X direction, the Y direction, and the Z direction. So you may be thinking, what does that mean? So that is you have one pointed this way, one pointed up and down, and one pointed kind of diagonally, okay? So within each of those, you have a whole bunch of fluid, and at the very bottom, you have a cupula that's, again, dealing with hair cells. So when the fluid moves, like if you move forward really quickly, that fluid is going to move forward also, but it takes a second for it to happen. And then the direction that it moves, depending on which canal it's in, is going to tell your brain what you're doing. So you can have your eyes closed and still move your head back and forth, and it's this that's telling you what's happening there. Okay, so between the semicircular canals and the utricle and the saccule, your brain can figure out where you are in space and what you're doing in space and how fast you're going. So 3 very important things. All based on the movement of fluid putting pressure on those hair cells, which is sort of like hearing. So we actually don't know probably this system will evolve first before hearing. Okay, so that is light and hearing. And we've talked about touch, we've talked about sight, we've talked about hearing. Let's talk about smell and taste. And then we'll be done with these. So, the smell. We have these things called chemoreceptors. They pick up chemicals. So our olfactory or our smell receptors are chemoreceptors. So they're sensitive to chemicals. Your smell is interesting because you undergo adaptation very quickly, but you can pick up a new smell very quickly. So if you put on some Axe body spray and then suddenly you can't smell it, so you think, I should put on some more. No. It's just that your brain has figured out that it's already there and it's adapted and it's done. So you don't have to put on 12 layers of Axe body spray. Trust me, we can all smell it. Everyone can smell it. But every time you smell a new smell, your brain figures out, oh, what is that? I need to pay attention. And this goes directly, so our nerves so here's our olfactory senses, okay? So here's the nose. We have air goes in here. Any chemical particles in the air are going to be picked up by those neurons, okay? And so the neuron's like, oh, I smell cooking. That's amazing. What is cooking? Am I going to eat that? I don't know. And so that immediately tells the olfactory bulb. So that is the part of the brain that's dealing with this, okay? And it says, hey, we smelled a smell. But the interesting thing, and my cats hear me talking about food, so they're freaking out. The interesting thing is this actually interacts very much with the limbic system. So we have very strong emotional associations with odors. So you could smell chocolate chip cookies, and, if it's not associated with any kind of emotion, it could just be, I smell cookies. Done. Am I going to eat the cookies? Am I not going to eat the cookies? Whatever. Or maybe you have an emotional sense where you're like, oh my God, my grandma made cookies. And she would bring them to me when I was sick and whatever. And then all of a sudden, smelling cookies makes you happy, which is a weird combination. Not that cookies making you happy, but the idea that a chemical can affect your emotions is kind of weird. But the reason why, so there's a lot of reasons for this. So we need to be able to smell food that we know is going to make us sick. So if, like a rotting smell if it makes you gag, that's actually a good thing because it's your brain is telling you, don't eat that, you could die, stupid, and so you don't eat it. Okay. Another really big reasons in mammals is that we smell mates even if we don't realize we're smelling it. Okay, so we can tell when somebody is ovulating. We can tell when, and this is usually. There's a bunch of studies where they have people go and work out really hard, and then they sweat into these t-shirts, and then they collect the t-shirts, and then they have people decide, do you like which of these 20 men that just sweated all over these t-shirts? Rank the t-shirts. Like, tell me which ones are the best smelling, which ones are the worst smelling. And there's all these weird associations, like men can tell when women are ovulating, women like men that smell more like their dads, or whatever weird association you have. There's a lot of studies out there like that we won't go into a lot of details, but it's interesting. So there's a lot of associations between smell and emotional state. We also, if you've ever had a cold, like my sinuses are stuffed up like all the friggin' time. It's so frustrating. And it's frustrating because it makes flavors taste less flavorful to me. So a lot of times when we are appreciating food, we smell, our sense of smell is a lot more sensitive than our sense of taste. And so we smell our food, even if we aren't doing it on purpose, we smell it before we taste it. And so when that's what's happening there is that chemical is, whatever that food is releasing the same flavor particles or flavor chemicals into the air. Your nose is picking them up first and then letting you kind of pre-taste the food through your olfactory bulb first. So a disease state, if you don't have any, for whatever reason, if you have anosmia, that's the loss of a sense of smell. This can be temporary if you have a cold, or it can be some sort of permanent state if you have a disease state, like a tumor in your olfactory bulb or something. Okay, so how does taste work? Taste works very similarly. It's another place where you have chemical signals activating chemoreceptors. Okay, these are on the tongue. They're also on the roof of the mouth, in the pharynx, which remember is in your throat, and there's actually some in our lungs. So there are bitter taste receptors in our lungs. For some reason, that makes our lungs open up a little bit. So a lot of times, a lot of very early asthma medication was actually to have patients smell something really bitter, like coffee, and that would actually decrease asthma symptoms. Albuterol is better, so don't just rely on coffee. Okay, so we can taste things for a long time. If you are eating something like red meat, a lot of times the fat in red meat actually covers up these channels that we need. These channels are full of so many taste buds. If you're eating something that kind of coats the top of this, it'll start to taste less. It'll taste less. You'll be able to taste less of it over time. And so when they talk about palate cleansers, what that means is you're then so like red wine pairs well with red meat because the tannins in the red wine wash that layer of fat off, and then you can taste your steak again in a better way, or you can taste it better. In the case, another palate cleanser, if you're talking about smell, is if you're testing perfumes, they have you smell coffee in between each time because it kind of freshens your senses because it's such a different smell than a perfume. Okay, let's see what else. So you'll notice this weird invagination or this indentation that goes on. The more surface area you have on something, the more receptors you can have. So that means we have a lot of taste receptors. If each one of those little buds on your tongue has a bunch of these all over it, that means you have a whole lot of taste receptors all over your tongue. You may have seen when you were a kid this idea that, oh, you can only taste bitters in the back of your tongue and sours. No, this is completely false. So our tongue has all of these taste receptors all over the entire thing, okay? There's also umami taste receptors, which are kind of this, they taste proteins, and you taste it if you're eating potatoes or mushrooms or steak. There's a certain taste that you can't describe as being sweet, salty, sour, or bitter, and that's umami. Chefs have known about this for years and years and years. Scientists only appreciated it about 15 years ago. Okay, so why do we taste what we taste? Sweet means sugar, so calories. So the reason why a lot of people have a sweet tooth is because when we were, evolutionarily developing, it was very, very rare to find things that were sweets. But if you found berries or something and you ate a whole bunch of them, then they didn't kill you, then you had a whole bunch of sugar, which means you had a whole lot of really easily accessible energy. Okay. On the other hand, sour and bitter so sour means acids, and bitter means bases. And in either case, if you are just in the wild eating these things could be toxic. Okay, so a lot of plant poisons are bitters. And so that's a lot of the reason why. So kids are much more sensitive to poisons because they're smaller. Literally, that's all. And so a lot of kids hate broccoli. They hate Brussels sprouts, other vegetables that are kind of bitter. But as you become adults, maybe you become to appreciate them more. And the reason why is you have less bitter receptors as you get older. They kind of recede a little bit. And so you can taste the other flavors in those vegetables. So if a kid is telling you they really hate broccoli, they're not just being pains in the butt because they want chocolate cake. They literally, it just tastes really bad to them. Salty. So we have become we've come to appreciate things that are sour and bitter. I'm thinking about beers in this case especially, but so a lot of times if something is really bitter, it could be telling you it's toxic. If it's really sour, it could be telling you it's something that's decayed. And so those are things to pay attention to. Salty means there's minerals present like sodium and chloride, which we need for our blood osmolality and for water balance and also for our neurons. So salty, a lot of times you could crave salt, and that's because literally you need to change your especially if you've been working out a lot and only drinking water, where you're sweating out a lot of salt, you need to take those salts back in, so you might crave something salty after that. And then umami again means is the flavor for protein, which is what am I trying to say? It's again telling you that something is nutritious, basically. These receptors are being regrown a lot. Oh yeah, so the connection to the limbic system may cause us to seek out certain foods and avoid others. So what that's saying is sometimes we stress eat. So we have food that we eat when we are stressed in certain ways. So when I have a really bad day, I want a beer and I want chocolate cake. So why do I want those things? Well, chocolate cake has got a lot of sweets, that's a lot of calories, and it's just kind of this homey feel to it. And then the beer has some bitter in it, and it's just, there's something, that's just, it's attached to a part of my brain that says, this makes me feel good. It doesn't make me feel nutritionally good, but it makes me feel emotionally good, which is weird. That's, no other animal eats for emotions, but humans do because our taste and our smell are so closely associated with the limbic system or the emotional system. We're weird animals. It's fine. Okay, so we talked about anosmia, or what is it called? So yeah, anosmia is the loss of smell. We know blindness is the loss of sight, and deafness is the loss of hearing. So there is the loss of taste, and that is, let's make this go away. So the impairment of taste is dysgeunia. This could be, again, this just like anosmia, this could be temporary, like if you're pregnant or on certain drugs. It could be due to a damaged nerve, okay? There's also aguisia, okay? This is a complete loss of taste, so it's not just things are off, it's just you can't taste. A lot of times this is either genetic or it's caused by a head injury, like a concussion where you're hitting the front of the limbic system, the front of the brain, which can be, usually could be recovered, but not always. There is something called the Gourmand syndrome, which is an obsession with very, very fine foods. This is actually, it's not a good thing. So if you know someone who only eats the finest food, they could have a stroke or a brain lesion, which is causing them to seek out novel things to try and appease their limbic system in a way that is not going to happen. Okay, so that's the senses. See you later. Bye.